So good afternoon, everyone.
Good afternoon, sir.
Good
Good,
afternoon,
af-
sir.
Good afternoon, sir.
good afternoon. So today we'll be dealing about the polyamines. So today we'll discuss what is polyamines, what are the different types of polyamines, its function, and how it is synthesized, and how it is removed from our body. What is the different biomedical importance? So first of all, it is by name itself we can say polyamines means many amines. It contains the many amines, the molecules having the many amines, that is known as the polyamines. Example, you can see the spermidine, spermine, and some of the books is mentioned the putrescine. Putrescine also the polyamines, but these are the two main, spermidine and spermine. The name is given spermidine and spermine because they have identified first time these polyamines in the semen. They have identified in the sample that is in the semen, so that's why the name is given spermidine and sperm. So the precursor for the polyamines, it is known as the substance that is a ornithine. Ornithine is a substance which act as a precursor for the biosynthesis of polyamines. So like in the bacteria polyamines and the mammalian polyamines, they are synthesized from the ornithine. So ornithine is a precursor for polyamines. It also requires the another molecules that is known as the methionine. The active form of methionine is required. Ornithine plus we need the another molecules that is a methionine. So these two molecules are required, but these are the precursor. Ornithine is the precursor from that the polyamines are synthesized. But for the synthesis, it also requires the active methionines. That is a S-adenosine, S-adenosyl methionine is required. In the normal human, it can synthesize approximately 0.5 n mole of the spermine per day. 0.5 n mole of spermine per day. So you have to remember is ornithine is a precursor. Plus, we also need the another molecules that is methionine. It is known as active methionine. S-adenosyl methionine is required for the synthesis of polyamines, that is spermine and spermidine. So how it is synthesized? What is the synthesis of polyamines? So as I told, the first precursor is the ornithine. From ornithine, it synthesizes first the putrescine. So how putrescine is synthesized from the ornithine? The enzyme that catalyzes the reaction is known as ornithine decarboxylase. Ornithine decarboxylase. So this ornithine decarboxylase causes the decarboxylation of ornithine to form the putrescine. Okay. Ornithine decarboxylase. This enzyme causes decarboxylation of ornithine and to form the putrescine. So putrescine is formed from the ornithine by the decarboxylation process, which is catalyzed by the enzyme known as ornithine decarboxylase. Now this putrescine, it forms the spermidine. From the putrescine, it forms the spermidine. So how it forms the spermidine is that, so putrescine combine or react with the decarboxylated S-adenosyl methionine. Just you remember, putrescine combined with the decarboxylated S-adenosyl methionine, and it forms the spermidine with the help of enzyme known as spermidine synthase. Spermidine synthase is helped to form the spermidine from the putrescine plus decarboxylated S-adenosyl methionine. So now how this decarboxylated S-adenosyl methionine is formed? So it is formed through active methionine with the help of the enzyme known as adenosyl methionine decarboxylase. Okay. Active methionine undergoes the decarboxylation with the help of the enzyme known as S-adenosyl methionine decarboxylase. S-adenosyl methionine decarboxylase. So the function of this decarboxylase enzyme is to remove the carbon dioxide. Molecule of carbon dioxide is removed from the active methionine and it forms the decarboxylated S-adenosyl methionine, which combined with the putrescine with the help of enzyme known as spermidine synthase to form the spermidine. Okay, so again I will repeat the same thing. So this ornithine undergoes decarboxylation with the help of enzyme ornithine decarboxylase to form the putrescine. Now this putrescine react with decarboxylated acetylcysteine methionine to form the spermidine with the help of spermidine synthase. This decarboxylated acetylcysteine methionine is formed from the active methionine with the help of enzyme known as acetylcysteine methionine decarboxylase, which causes the decarboxylation of active methionines and it forms the decarboxylated acetylcysteine methionine. This is the one polyamines which is formed from the ornithine. It forms the putrescine, then polyamines, that is a spermidine. Now from this spermidine it forms the spermine by the enzyme spermine synthase, and this also requires the presence of another molecules of decarboxylated S-adenosyl methionine, and it forms the spermine. From the spermidine, okay, from the spermidine it forms the spermine, and that also requires the decarboxylated S-adenosyl methionine, and the reaction is catalyzed by enzyme known as spermine synthase. So for more clarification, you can see here so how it forms. So first you will see this one. This is the precursor, that is a ornithine. Decarboxylation, so carbon dioxide is removed, it forms the putrescine. So this putrescine it forms the spermidine by combining with the decarboxylated adenosyl methionine, and from this spermidine it forms the spermine. This reaction also combined with the decarboxylated S-adenosyl methionine with the help of enzyme known as spermine. So like this, it's very simple. Simply it can synthesize the polyamines. So these are two main polyamines, spermidine and the spermine, which is synthesized from the ornithine. Okay, so ornithine decarboxylase, the first enzyme, then spermidine synthase and spermine synthase. Only the three enzymes. Plus we need one additional enzyme that is required for the formation of decarboxylated S-adenosyl methionine from the active methionine. So this is the enzyme S-adenosyl methionine decarboxylase. So here also you can see the molecules of carbon dioxide is removed from the S-adenosyl methionine, that is the active methionine, and it forms the decarboxylated S-adenosyl methionine. Here you can see this plus sign. This plus sign indicates that putrescine increases the synthesis of decarboxylated S-adenosyl methionine. So that means it increases the formation of decarboxylated S-adenosyl methionine. If there is a putrescine, the concentration of putrescine is increased, that causes activation of this enzyme. S-adenosyl methionine decarboxylase. So if this enzyme is activated, this causes the decarboxylation of S-adenosyl methionine, and it forms the decarboxylated S-adenosyl methionine, which is required for the formation of the spermine and the spermidine. But this minus, you can see minus sign, this indicates that if there is a more amount of decarboxylated S-adenosyl methionine, this will inhibit this enzyme. Okay, inhibits the S-adenosyl methionine decarboxylase. That means this indicates that there is enough amount of decarboxylated S-adenosyl methionines. There is no need of further breakdown of S-adenosyl methionine. So this enzyme will be inhibited and this reaction will not take place. This reaction will not take place because this enzyme will be inhibited by the product itself. This is another feedback inhibition, feedback. So this product itself will inhibit the decarboxylase enzyme. So S-adenosyl methionine will not break down into the decarboxylated S-adenosyl methionine. So it's a very simple synthesis, you can say. Mainly in the 3 steps, polyamines are synthesized. So you have to remember these 3 enzymes plus these additional enzymes. So for the regulation of polyamine synthesis, this is the main enzyme, ornithine decarboxylase, and plus we have the S-adenosyl methionine decarboxylase. This is the second enzyme. To that you can regulate. If this enzyme is also inhibited, the decarboxylated S-adenosyl methionine will not not available, so there is no formation of the spermidine and spermine. So these are the two main enzymes to that you can control it and inhibits or increase the rate of formation of the polyamines. So how it is regulated? So you can see, as I told before, ornithine decarboxylase. This is a main enzymes which activity is increased by growth hormones, corticosteroids, testosterone, epidermal growth factors. So these are the few hormones which activates the activity of or increase the activity of the ornithine decarboxylase. So from the ornithine it forms the putrescine, and this enzyme causes the decarboxylation of ornithine to form the putrescines. So this reaction is increased. The activity of this enzyme is increased by growth hormones, corticosteroid, testosterone, epidermal growth factors. The second enzyme, as I said, is S-adenosyl methionine decarboxylase. So the activity is inhibited by the product itself, as I said before, decarboxylated S-adenosyl methionine, and activated by the putrescine. If there is a more amount of putrescine is formed from the ornithine, then this activates the S-adenosyl methionine decarboxylase enzyme and help in the formation of polyamines like spermidine and spermine. So how these polyamines are breakdown? How it is degraded? What is the degradation of the polyamines? So the main enzyme which involved in the degradation of polyamines is known as the oxidase. Oxidase is the main enzymes which causes the degradation of the polyamines, and this is known as the polyamines oxidase. This is present in the liver peroxisome, okay, present in the liver peroxisomes, and oxidizes the spermine to spermidine. Just reverse. Before it was from the putrescine, it forms the spermidine, then spermine. Now in the degradation process, from the spermine it forms the spermidine, then the putrescine, then finally it forms the carbon dioxide. So this oxidase, polyamines oxidase, is causes the oxidation of oxidizes the spermine to spermidine. Now this spermidine, by again the same enzymes, polyamine oxidase, it causes the formation of the putrescine by the same enzymes. Here also the same enzyme, and down also from the spermidine it forms the putrescine by the same enzyme, that is a polyamine oxidase, and 2 forms the putrescine. Now finally this putrescine converted into the ammonia and the carbon dioxide. These are the two products which is formed, ammonia and the carbon dioxide. And the putrescine and spermidine are excreted through the urine. And how it is excreted? It will undergo the acetylations, and acetylated derivatives are excreted out through the end. This is the major portions. It will be causes the acetylation, undergo the process of acetylations, and these acetylated derivatives are being excreted out. Okay, in this way the polyamines are undergo the degradations. So you have to remember is the oxidase enzymes, which is a polyamines oxidase present in the liver peroxisome, which oxidize the spermine first to spermidine. Secondly, this spermidine converted into putrescine by the same enzyme, that is a polyamines oxidase, and finally this putrescine converted into the ammonia and the carbon dioxides, and this major portion of this putrescine and spermidines excreted through the urine after the acetylations. Acetyl group will be added, and these acetylated derivatives will be easily excreted out through the urine. So in this way, there is degradation and also the excretion of the polyamines from our body. In the simple way, you can see in the diagram way. So this spermine with the addition of the molecules of oxygen is formed the spermidine, that's putrescine, and finally carbon dioxide and ammonia. Polyamines oxidase, and you can see here H2O2 is also released during the degradation process. H2O2 is released, plus another molecules that is known as the beta amino propione aldehyde. Beta amino propione aldehyde here also, and here also it is released. Okay, but finally it is forms the carbon dioxide and ammonia. These are the compound which is also released in the formation or oxidation of this spermine and spermidine. But enzyme is the only one that is oxidase, known as the polyamine oxidase. So if you see the natural structure of the polyamines, you can mainly it is the polymers of diamminobutane and the diaminopropane. These are the two compounds which is mainly present in the polyamines, that is spermine and spermidine, diaminobutane, diaminopropane. So what this putrescine contains? It contains the 1,4-diaminobutane and this spermidine. This putrescine, that means 1,4-diaminobutane plus 1,3-diaminopropane is contained. That is in the spermidine. In the spermine, two molecules of, you can say, 1,3-diaminopropane, 1,3-diaminopropane plus putrescine. So the number of amino group is increased, like in the spermidine and the spermine. Okay, so these are the natural structure which are the polymer of the diaminobutane and diaminopropane. So this is the natural structure of polyamides. So what is the function of the polyamides? Why it is required for our body? If there is no function, then we have to study this. Why there is a synthesis? So you can see these are the cations. Okay, these polyamides are the cations involved in the stabilization of the anionic structures. Remember, it is a cationic substance which involved in the stabilization of anionic structures like DNA, ribosomes, subcellular organelles, and the membrane. Okay, which you can say that simplifies the nucleic acids. The structure of the nucleic acid, you can say DNA or RNA, ribosomes, subcellular organelles, and the membrane. This is also involved in the maintenance of the structure of the tRNA. If you remember the structure of tRNA, it is a clover leaf structure. The tRNA having the structure that is not a clover leaf structure, and that structure is also, this polyamine is also involved in the maintenance of that structures. And they are also required for the packaging of the bacteriophage DNA. So, and we also stimulate the nucleic acids and the protein biosynthesis. So biosynthesis of the protein and nucleic acid is stimulated by the polyamines. These polyamines also required for the growth or the cell divisions. So what you have to remember here is it stabilizes the anionic structure like DNA, ribosomes, subcellular organelles, and the membrane, involved in the maintenance of the tRNA structure that is known as the clover leaf structures, also required for the packaging of bacteriophage DNA. It stimulates the biosynthesis of the protein and the nucleic acids and required for the growth or cell divisions. Cell growth and cell divisions. So these are the few functions, and you can see here, like if you want to culture the mammalian cell and the bacterial cells. So these polyamines act as growth factors. It is required as the growth factors for the cells. For the culturing the mammalian cells and the bacterial cells, these polyamines require the growth factors. And these polyamines also act as some of the markers. You can see here the marker of the tumor cell distractions. The polyamines known as the spermidine, and this act as the best marker of tumor cell distractions. So act as a marker. That is also one of the functions of the polyamine spermidine, best marker of tumor cell distractions. In the pharmacological dose, polyamines found to be hypothermic and hypotensive. So these are also the function, hypothermic and the hypotensive. They act as inhibitors of the enzymes. Inhibitor of the enzymes that is known as the protein kinase. So it acts as an inhibitor of the protein kinase enzymes. So these are the few functions. Even the markers, it can act as a hypothermic and hypotensive in the pharmacological doses, and require for growth factors for the culturing of bacterial cell and mammalian cell, and inhibitor of the few enzymes that is protein kinase. So what is the medical importance of polyamines. So polyamines are required for the cell division. So the inhibitor of the polyamines biosynthesis can be used as a anti-cancer drugs. As in the function network, it is required for the cell divisions, cell growth. So the inhibitors of the polyamine biosynthesis inhibitors can be used as the anti-cancer drugs. Okay, because the number of polyamines is more in the cancer cells, because it is required for the cell division and the cell growth. So in the cancer cells, it is uncontrolled growth, uncontrolled cell division. That means the number of polyamines required is more. So those substances which can inhibit the polyamine biosynthesis can use as anti-cancer drugs. One example you can take is the DFMO, difluoromethylornithine, difluoromethylornithine. This is the inhibitor of this enzyme, ornithine decarboxylase, which is the first enzyme used in the polyamines biosynthesis. So this can be used as the anti-cancer drug. Anti-cancer drugs can be used. This DFMO, ornithine decarboxylase enzyme inhibited by the DFMO, difluoromethylornithine. This DFMO is also used to cure the certain protozoal and the parasitic infections. Okay, the few protozoal and parasitic infections can be cured by the difluoromethylornithine. So one interesting thing you can observe, like the polyamines excretion. So excretion of polyamines in the urine is increased in the most of the cancer. In the cancer patient, if you will test for the polyamines in the urine sample of the cancer patient, the polyamines will be more. You can get a more amount of polyamines in the urine of the cancer patient. In most of the cancer, like leukemia, ovarian cancer, lung cancer. So in that case, you can find the polyamines in the urine sample of the cancer patient, leukemia, ovarian cancer, lung cancers. So the polyamines concentration is more in the cancer cells. The reason is it is required for the cell division and the cell growth. So polyamines required for the cell growth and cell divisions. That's why these cancers here are the uncontrolled cell division and the cell growth. That's why the concentration of polyamines more in the cancer cells. That means in the cancer patient. So if you check for the concentration of polyamines in the urine sample of cancer patient, you will get more amount of polyamines in the urine. So presence of spermidine is used to identify a sperm in the substrate victim. Even this can use for the victim. Okay, so in that you can presence of the spermidine in the sperm of that, you can the sperm you can identify the spermidine or the presence of spermidine in that the victim. But nowadays this is not used. So spermidine, as I said before, it is the best marker for the tumor cell destruction. Similarly, the putrescine is also the best marker for the cell proliferations. So remember, marker of tumor cell destruction, that is spermidine, and the putrescine is the best marker of the cell proliferations. So these two can be used as the marker for the tumor cell and the cell proliferations. So these are the medical importance of polyamines. So you have any doubt? This is regarding the polyamines, how it is synthesized, what is the function of the polyamines, how it is integrated, degradation of the polyamines, and the medical importance, why it is, how it is clinically related. Okay, so this is all regarding the polyamines. So we can see the few MCQ related to today's topic. So best marker of tumor cell destruction is which of the following A, B, C, D. You can find out the like putrescine, spermidine, none of the above. So take the which one is the right answer. Then second is the best marker for the cell proliferations. That is a putrescine, asparmine, spermidine, or none of the above. The third MCQ, which of the following is precursor of the polyamines, like methionine, or it is ornithine, or it is citrulline, or it is a glutamate. What is the right answer? You have to take it, either A, B, C, or D. The fourth MCQ, that is ornithine decarboxylase activity, is inhibited by all of the following except. So out of these four, the three is the right answer and one is the wrong answer, which is not activated the activity of ornithine decarboxylase. So you have to find out that one. Similarly, you have the some short questions like write down the synthesis and regulation of the polyamines. Write down the synthesis and regulation of the polyamines. Second one, the list out the function of the polyamines. What is the function of the polyamines? The major function you can list, not the all, not the major function you can add here. And write down the degradation process of the polyamines. So how the polyamines are degraded and excreted out from our body. So that you have to answer. So from this topic, these are the possible questions you can have, and also the MCQ. One more MCQ you can make, like which enzyme is regulated, you can say, in the biosynthesis of the polyamines. What are the enzymes which involve in the regulation of the polyamines? So you can say ornithine decarboxylase and in this one, acetylinosyl methionine decarboxylase. These are three enzymes which is involved, but main is the ornithine decarboxylase. So this is regarding the ornithine, how it is synthesized, what is the structures, and what are the precursor of the polyamines we have discussed. So just I will explain once again the synthesis, how it is synthesized, just for the division. Like ornithine, it forms the putrescine because it undergoes the decarboxylations by the ornithine decarboxylase, and it also requires the vitamin B6. Paradoxal phosphate is required for this enzyme, ornithine decarboxylase. Then this putrescine react with the decarboxylated acetyl methionine to forms the spermidine with the help of enzyme spermidine synthase. Now this spermidine again it is combined or react with the decarboxylated acetyl methionine to form the spermine with the help of spermine synthase. And these polyamines are synthesized. So how this decarboxylated acetyl methionine is formed? It is formed from the active methionine that is known as S-adenosyl methionine or active methionine, or you can say S-adenosyl methionine. This also undergoes the decarboxylation and there is removal of the carbon dioxide by the enzyme acetylinosyl methionine decarboxylase, and because this is a decarboxylase enzyme which is mainly contains the pyridoxal phosphate. It requires the coenzyme that is a pyridoxal phosphate which is required for the decarboxylase enzyme, and it forms the decarboxylated acetylinosyl methionine. So in this way there is a synthesis of polyamines. And how it is regulated, like this putrescine activates the acetylinosyl methionine decarboxylase, and it is inhibited by the decarboxylated S-adenosyl methionine because this acts as a feedback relations. The product is to go and inhibit the S-adenosyl methionine decarboxylase. So in this way it is synthesized. And one more thing, how it is break down, this one you have to remember is the oxidase enzymes, polyamines oxidase. It is the enzymes which causes the breakdown of polyamines from spermine. It forms the spermidine, then it forms the putrescine, and this putrescine is finally converted into carbon dioxide and ammonia. And there is a sum of the product which is formed in between, that is beta amino propanaldehyde, and these are the undergoes the acetylations, and these acetylated derivatives are excreted through the urine. Okay, excreted through the urine. So these are the relation how it is. polyamines. So do you have any doubts? Any doubt regarding the polyamines? No doubt, and thank you for today.